Okay, yeah, so like Chris said, I'm filling in for Lily on this. My role in the plankton sampling is the net thrower and the hauler. So I am involved in the whole sampling process, but I'll talk a little bit about the analysis as well, which Lily has been doing. So these are the project objectives. I actually meant to ask Chris, how far through this program are we? 1 year? 1 year of 3 years?
We've done 2 seasons, and this is sort of taking a more rigorous approach. So this coming season is a much more rigorous approach.
Far more rigorous.
Sampling, which we hope will carry on for another 2 or 3 years.
Yeah, cool. So we are aiming to identify the zooplankton and determining basically the mechanisms that are driving the fish shoaling activities and the link between these fish shoals and the seabirds. So there's many different components to this study, but we're primarily looking at the interaction between the seabirds, the zooplankton and the fish through a range of field and lab techniques. And the methods that are highlighted in red are the ones that I'm going to discuss today. So this is kind of our sampling triangle that we go out in. We have mainly been using James's boat up until now, but we'll also start to use the University of Auckland boat starting in January. And we've usually just done 1-day trips, but we're going to be throwing in some 3-day trips as well, where we'll do sort of continuous sampling over those 3 days. So we keep quite detailed records of the workups that we encounter and include the GPS position, the approximate size of the workup, the fish and seabird species present, and whether there's any other additional megafauna such as whales and dolphins. Where possible, we aim to take really high resolution photos from aboard the boat, which provides further insight into the feeding behaviours and the general activity around the workups. So a zooplankton sampling net is towed horizontally behind the boat to collect the zooplankton. And these are then preserved in ethanol, where they go to the Lee Marine Lab to be photographed and measured and analysed. And in the coming months, we aim to determine the energy content of zooplankton with a bomb calorimeter, which is one of our new methods that we have not done yet, but that will be cool. So to see the energy content of the zooplankton that we sample. And we will be sampling zooplankton both inside those fish schools and outside to act as a control. So here's just a couple of the zooplankton that have been identified so far. So they get identified into 7 different groups. And these are photographs taken through the microscope at the Lee Marine Lab. And as you can see, they range considerably in size and shape, from the krill up there in the right-hand corner to these fish eggs here. So that's 4 of the 7 groups. And those are the other 3. And so in addition to those easily recognisable krill and salps, we often get little fish, juvenile fish as well. And so because seabirds both feed on and in association with the fish, we sample the muscle tissue and the stomach contents of these fish to determine what they have also been feeding on and the energy content of their prey as well. So that's one of our new methods we're going to be looking at. And so Chris kind of talked about this, but yeah, we use a floating camera rig to get a better idea of sort of the species that are in the fish shoal and whether there's krill present as well. And this is a camera that we have pointing directly at the mouth of the net, and so we can use it to look at any avoidance behaviour, whether those zooplankton are avoiding the net in any way. And it also creates some really cool, quite mesmerising videos, which we didn't get to see one of, but they are very cool. And we look at a number of environmental variables during sampling. So these parameters, along with the GPS coordinates, are really useful in assessing why the workups may occur where they do. And then Lily will be doing some really complicated statistics to determine whether there's any differences in the zooplankton composition both inside and outside those fish schools, and whether the zooplankton consumed by fish differs to the zooplankton that we connect during the net sampling. And further analysis will seek to determine associations between zooplankton, fish species, seabirds and environmental parameters. And one thing that we would love to include is more drone footage, so you can get a far better picture of how big a fish school is from above than you can from, at sea level on the boat. So we'd love to include some more drone footage in future, hopefully. And we'll be comparing the seabird regurgitations that Chris spoke about earlier with the zooplankton samples that we collect as well. Thanks. Some